Hello guys, my name is Kumar Yogeshwaran. I'm from Group 16, 5th course, 6th faculty, Kharkiv National Medical University. And today I'm going to be talking about gene mutation, one of the interesting topics in medical genetics. So let's dive right into it without wasting any time. So before talking about gene mutation, let's get a broad or a narrow idea about what is mutation actually. So mutation, we can say it as any sudden change occurring in hereditary material. And these mutations can be harmful, they can be beneficial, or they can be neutral. Like doesn't show any function at all. They're just happening and we don't even notice them. Like they don't show any function. So that's why they stay neutral. That's what neutral means. So yeah. And so we have two types of mutations. We have gene mutation and we have chromosomal mutation. So as today my topic is about gene mutation, so I'll be talking only about gene mutation. So let's talk about gene mutation. So what is gene mutation? Gene mutation is mutation or series of mutations occurring in the polynucleotide sequence of a gene that changes the function of it. That changes the function of it, of the DNA, or the function that the polynucleotide sequence was prepared to do. The mutation changes it. That's what gene mutation is really known to be. So, or we can say it as, in a definition, we can say it as any sudden alteration in the sequence of nucleotide in DNA. This change in DNA will reflect the change in the phenotype. So what is a phenotype? We say, if you know the basics of genetics, phenotype is nothing but the observable characteristics of an individual, like which the characteristics which we see in a person. Okay, so those that is called as a phenotype. So whatever mutation that takes place in the body, we can see it in the phenotype. And these changes, these mutations, they can occur in a single nucleotide pair or on larger segment of a chromosome. Depends upon the stages of the mutation. And one of the important points, mutations is one of the main factors that contributes to our evolution. So as evolution takes place in human beings, we get adapted to certain things, certain like temperatures, for example, certain climates. These things happen because our genes are programmed in that way, or they are changed in that way so that we adapt to the particular environment or the particular situation. So this is possible only when the DNA or the gene which performs a particular function, they have to change according to the environment. So it's also a type of mutation, we can say. And we can say that it contributes, like it has a major role or major contribution to our evolution. So we can say like mutations, they can be occurred spontaneously or they can be induced. So what do we mean by spontaneous mutations and induced mutation? So spontaneous mutations are the mutations that occur by natural causes. They are not induced. They occur by our how to say, spontaneous mutations are those which arise naturally, not through the action of a mutagenic agent. They may arise through, like we can say, for example, errors in DNA replication or spontaneous alteration of a nucleotide within the existing DNA molecule. Like there is no induced effect. The body by themselves, they delete or they change certain parts of a DNA which results in mutation. It's not any external factor that causes the mutation. Those type of mutations are called as spontaneous mutations. So spontaneous mutations we can see are of like three types. We have Tautomerism, we have depurination, and we have deamination. So what is tautomerism? In tautomerism, a base is changed by repositioning of a hydrogen atom, altering the hydrogen atom pattern of that bases, resulting in incorrect base pairing replication. That is tautomerism. Like a base is changed by repositioning an hydrogen atom. We know that each base in the DNA molecule, they are joined by hydrogen atom. So those bonds are repositioned with different type of bases, which causes incorrect base pairings. So incorrect base pairing results to mutation. They can give rise to different functions. They can be neutral. They can be harmful. We don't know. Those type of repositioning of hydrogen atom base pairs are called as tautomerism. And another we have is depurination. Depurination, we know that in a DNA molecule we have purines and pyrimidines. So in depurination, there is a removal of purine base, like adenosine or guanine, which forms a puremic site. That's why we call it as depurination. The third spontaneous mutation part we have is deamination. Deamination is because of hydrolysis. Hydrolysis changes the normal base to a typical base containing keto group in place of original amino acid group. So when with the DNA, when the DNA for the transcription takes place, the DNA gives us to mRNA. mRNA then translates into giving of amino acids. But in place of original amino acid, there is a keto group present because of hydrolysis takes place in typical base, which makes them to contain keto group in place of amino acids. This is called as deamination. And the second type of mutation, other than spontaneous mutation we were talking about, is induced mutation. So what is induced mutation? And like induced mutation is because an organism is exposed to a variety of physical, chemical, and biological agents capable of causing damage to its genetic material. Okay, like physical and chemical substance or biological substance that results in a mutation in an individual. So the physical mutagens which can cause mutations are it's like radiation or heat. In radiation we have two types of radiation. We have ionizing radiation and we have non-ionizing radiation. And then we have heat. Like heat in the sense UV rays from the sun can cause mutation. Like for example in Australia, it's said that they have a hole in ozone layer over the atmosphere in Australia, because of which there is a direct UV ray among the population, which causes more skin cancers among the population. So skin cancer is also cancers are proliferating cells. They are gene related. So maybe this UV rays cause those genes to mutate, which results in proliferating of the cells, causing cancers, skin cancers among the population. So this is one of the example of physical mutation, heat-induced physical mutation. Okay. The second we have is chemical mutagens. In chemical mutagens, we have base analogs, alkylating agent, intercalating agents. Okay. So let's move. So as I said before, there are different effects of gene mutation. Some are beneficial, some are harmful, and some are neutral. The beneficial part of the gene mutation is they produce new version of protein which will be helpful for us, and which will be helpful for us and it will be helpful for our evolution, and because of which it will enhance our survival and our reproductive success. And maybe these beneficial gene mutation can also kill harmful genes that results in diseases in our body, like cancer, for example. These type of mutations are called as beneficial gene mutations. Are called as beneficial gene mutation process. Another one is harmful, and most of the gene mutation we are said to be harmful type of gene mutations, because always mutation, people when they hear mutation, their mind directly flows into harmful aspect of it. So no one thinks of it as a beneficial part. Some people they think of the beneficial part, but most often when you hear the word mutation, I bet we're not talking about X-Men here, so it's not good. It's really bad. So everybody thinks it's bad. So the harmful effects are protein malfunction, as we said before. Protein malfunction, a protein which has to perform a certain function will not perform the certain function, maybe it diverts from the certain function, which disturbs our homeostasis, and causes problems in our body. So we don't want that. So protein malfunction, and which is lethal to life, and it can lead to some genetic disorders. And another one is neutral. Neutral gene mutations are neither they provide harm for our body, and neither do they benefit our body. So these type of mutations are called as neutral mutations. So let's move on to our next slide. See what we have here. Yeah. So let's talk about types of gene mutation. So what are the types of gene mutation? Gene mutations can be described as can be categorized as point mutation, frame shift mutation, and base substitution mutation. So what is point mutation? Let's see. Point mutation is the most common type of mutation. It is also known as the base pair mutation. It shows change in the single nucleotide base pair. It can be like genetic transition, like that, which means that there's a replacement of one purine with another purine, like example guanine to adenine, adenine to guanine, or the replacement of a pyrimidine with another pyrimidine. And also there is genetic transversion. That means there is a replacement of purine with a pyrimidine, or there is a replacement with a pyrimidine with a purine. Okay, and point mutation can hence further be classified into silent mutation, missense mutation, and nonsense mutation. And we'll talk about these mutations one by one detailed. Okay, so let's see what is. Yeah, we said point mutation can be further categorized into three different type of mutations. One is silent mutation, one is missense mutation, and another one is nonsense mutation. So what is silent mutation? So we'll come back to this image later. Let's see this image. Like, this is the original DNA, which consists of the corresponding amino acid, which is leucine. So let's see, what does it mean, silent mutation? So there are codons in our body there are three pairs, and these three pairs together, when they combine, they give us an amino acid. And these amino acids perform a certain give rise to proteins these amino acids are part of proteins which perform a certain function in our body. So what it does is that it changes one codon of an amino acid with another codon, but when they change the codon each other, when they change it, the amino acid still remains the same. I'll repeat, like when there's a change of one codon for an amino acid to another codon, it results in the same amino acid. There is no change. That's why the name says it as silent mutation. Silent mutation, we can say it as the neutral type of mutation, which doesn't really benefit or cause any damage. It just changes an amino acid it changes the codon for the same amino acid. It doesn't really cause any. It doesn't really show any effects later on after the mutation has been taken place. That's why it's called a silent mutation. But we still characterize them into mutation because there is a change in the codon. There is change in the base pair. So when there is a change, what we said, when there is an alteration of the sequence, we call it as mutation. But it is a silent one, so we say it as silent mutation. Okay. After silent mutation, we have missense mutation. So what is missense mutation? So codon for one amino acid is changed into codon for another amino acid. See, silent mutation, codon for one amino acid changed into same amino acid, but in missense, it doesn't code for one the same. It changes the amino acid. When the codon, the base pair sequence like ATG, GAA, GCA, like ATG, like glutamic acid, GAA is glutamic acid. When it changes, the base pair change, it doesn't give rise to the same amino acid. It changes the amino acid, which causes mutation. The changed amino acid will perform different function or will not perform any function. So this type of mutation is called as missense. One amino acid is for one codon for one amino acid changed into codon for another amino acid. It's called as missense mutation, and this missense mutation is sometimes called as non-synonymous mutation. Okay. The third is nonsense mutation. Nonsense mutation. Mutation is nonsense, but all the sarcasm aside, what nonsense mutation is it like codon for one amino acid is changed into stop codon for translation. So there are particular codons which are called as a stop codon. Okay, these stop codons when there is translation process going on, these stop codons stops the translation, which means there is no more protein synthesis taking place, and these proteins will not then go perform any function. So in nonsense mutation, what happens is that the codon sequence is altered in such a way that it results in formation of stop codon, and this stop codon stops translation. So that's why it's called as nonsense mutation. Even the body doesn't like it. Like, who likes to stop protein synthesis? You have to grow, bro. So, and the next, after point mutation, we talked about point mutation, frame shift mutation, and base pair mutation, right? So we finished point mutation, and there are three categories. And the second we have is frame shift mutation. So what is frame shift mutation? This type of mutation occurs when an addition or loss of DNA bases change a gene reading frame. A reading frame. What is a reading frame? A reading frame consists of 3 bases, each code for one amino acid. That's a codon. That's the reading frame. I said like 3 codons, when they combine together, they form one amino acid. This amino acid gives rise to a protein, and they perform a function. So there's a change. There is an addition. The mutation in frame shift mutation is like there's an addition or a loss of DNA bases that changes the gene reading frame. In this mutation, the codons in these base pairs are changed, which results in change of amino acid. Yeah, and this frame shift mutation can further be categorized into insertion or deletion. So, what is insertion? As the name itself says, insertion means you add an codon. So let's see. First we have is TAA, the sequence. Let's see, there are three sequences: TAA, CTG, CAG, and GT. See, this is like in a sequence they are going. Okay. So when you insert this, after this, see, there is an insertion of another codon. In the codon, there's an insertion of one more base pair. What it does is that it makes this 3, then it counts this 3. After this, first we had the base pair as TAA, then we had CTG, then we had CAG, and then we had GT. Okay, 3 base pairs. After that, what happens is that the TAA remains same, but the CTG changes into CCT, and the CAG changes into GCA, and the GT changes into GGT. So what happens is that whichever amino acid they were coding before, it will not be the same. The first one is the same. The first TAA will be the same, but afterwards, whichever codon or the amino acid is following will change compared to the original sequence. This is called as the insertion. See, after the C in the 4th position, there's insertion of another C. This is called as insertion, addition of one more base pair. I hope you can understand. You can't see my pointer over here because I'm learning to use this app. So excuse me, but after TAA, you can see the base sequence TAA, that is CTG. But here there's insertion after the C, there's insertion of another C, which causes it to become CCT instead of CTG. So this will result in a different amino acid, which will result in a different function. So this is called as insertion type of mutation. So let's go. After insertion, we were talking about deletion. In deletion, what will happen? See the base. Let's see the original sequence. We have TAA, we have CTG, we have CAG, and we have GT. All right. Now what happens? There is a deletion of one or more base pairs. So in the deletion base pair, we can see that the TAA remains same, but the CTG, the T has been deleted, which will make the another base pair has to be CGC instead of CTG. It will become CGC, which will result in another amino acid because one of their base pair has been deleted. It's called a deletion mutation. It's simple. Insertion deletion is very simple. And now we have substitution mutation. Frame shift mutation we're talking about, and this is the third type. It's the substitution. It's like one or more base pairs are replaced by another base pairs. So what we see here is the TAA, CTG, CAG, and GT. But the GAT, the TGC, in the original sequence we have TGC, right? This TGC has been substituted with GAT because of mutation. When they have substituted with another frame, like what we call it as reading sequence, they're changed with another reading sequence. Instead of TGC has been removed. We can see here in the substitution mutation in the second sequence, the TGC is removed and GAT has been inserted. So this type of mutation is called as substitution mutation. You can see, because it has been replaced by another base pair. The original base pair has been replaced by another base pair. Okay. So let's move on. What do we have here is trinucleotide repeat expansion. This is another what we call it as another category apart from the frame shift mutation. It's called as a trinucleotide repeat expansion. What it really means is that increased repetition of base triplets that leads into faulty protein synthesis. Increased repetition of the base triplets results into faulty protein synthesis. It's called as the trinucleotide repeat expansion. Expansion means more. If we repeat something for a long time, the trinucleotide sequence has been expanded and it causes a faulty sequence, and it causes a faulty sequence which results into faulty protein synthesis, which will lead to default in our body. And this trinucleotide expansion can lead to different type of diseases, like we can see here as Huntington diseases, and that is which part of the triplet which has been repeated continuously is CAG. Here is the triplet. It's called as a CAG, and we can see it as Huntington's disease is a autosomal dominant disease. And second, we have a Fragile X syndrome, which is an X-linked recessive mode of inheritance. And the main trinucleotide sequence which has been repeated here is CGG. Okay, and the chromosome X has been main feature. And we have myotonic dystrophy in the third point. Myotonic dystrophy, it is autosomal dominant mode of inheritance, and the trinucleotide sequence which has been repeated over here is CTG. Okay, and one of the typical features of this myotonic dystrophy is cataracts and hair loss, we can see. And fourth we have is Frederick Ataxia. Frederick Ataxia is also autosomal recessive mode of inheritance, and the trinucleotide sequence which has been repeated over here is GAA. So these are like some of the examples of trinucleotide repeat expansion diseases. Then I'll say it again. It's because of triplets. It's because of base triplets that leads to faulty repetition of base triplets that results into faulty protein synthesis, and they cause these type of diseases. And these triplets are like CAG, CCG, CTG, GAA. These are some of the examples of those. Yeah. So let's move on to the next slide. What we have here is, yeah, let's talk about other types. We talked about point mutation, we talked about frame shift mutation, we talked about trinucleotide sequence expansion, and now it's time of talking about other type of mutations which didn't come in any of these categories, in the following categories. And these mutations we can say it as loss of function mutation, gain of function mutation, and we have splice mutation, and we have dominant negative mutation, and some other are also called as the forward mutation. We have backward mutation. So what is forward mutation? I forgot to mention over here. I forgot to write down over here. But there's another mutation we call it as the forward mutation. Forward mutation is like a genetic mutation from wild type to mutant, or evolution of new mutations from wild type allele is called as forward mutation. The forward mutation leads to the evolution of new traits in the population. Yeah, like forward mutation are the mutations which results in evolution. Okay, and we have another type of mutation is called as the backward mutation. Backward mutations is like is very rare, and it's like an unusual phenomenon in nature. It gives original phenotype back into the population by true back mutation or by the occurrence of secondary mutation. Like the mutation is not in a progressive way, and it's not very usually seen in the population. The backward mutation, like these are some. Then we have duplication. Duplication mutation are certain type of mutations when some base or a base is duplicated in a gene sequence. These base pairs are duplicated in a base sequence. These are called as duplication mutations. And let me check if I have missed something. Yeah, another type of mutation is inversion mutation, when some gene sequences are inverted and inserted back into the original sequence, as also called as, like, if there's there's a triplet base called which is said to be TAA, they're inverted and they become AAT. This is an example of inversion mutation. And let's see if I have missed any point over here. Okay, we talked about frame shift mutation, we talked about point mutation. So let's see. So yeah, these are the main type of mutations which are which really make which are mostly studied in this gene mutation topics. There are harmful effects of mutations. There are like, imagine making a random change in a complicated machine, like such as a car engine. The chance that the random change would improve the function of the car is very small. The change is far more likely to result in a car that does not run well, or perhaps does not run at all. By the same token, any random change in a gene's DNA is likely to result in a protein that does not function normally or may not function at all. Such mutations are most likely to be harmful. Harmful mutations may cause like genetic disorders or cancers. And what we have here is genetic disorders. So genetic disorders are like which are caused by mutation of a single gene. This single gene can lead to missing or malformed proteins that can lead to different kind of genetic disorders. Like a human example of it is cystic fibrosis, where a mutation in a single gene causes the body to produce thick, sticky mucus that clogs the lungs and blocks the ducts in digestive organs. And the second is cancer. Cancer is a disease in which cell grows out of control and from abnormal masses of cells. It is generally caused by mutation in genes that regulate the cell cycle. Because of mutation, cells with damaged DNA damaged DNA, they are allowed to divide vigorously, and they multiply in such a way that they cause cancers. And these cancer genes can also be inherited, which means they can be passed down. So what I'm making a point is that even a small change in a gene can be passed on to generations and can cause problems. That's what I am trying to say here. And there are also like some beneficial effects I wanted to say, like there's unique mutation is found in people in small town Italy, where the people are mutated in such a way that they are resistant to atherosclerosis. Yeah, you heard me right. They are resistant to. There is a town in Italy where they mutated in such a way that they are resistant to atherosclerosis. How cool is that? Yeah. So, with this, I would like to conclude my lecture, and thank you for paying attention and listening to me. I hope you liked my presentation, and thank you, sir, for providing me with this opportunity. And thank you, and hope you have a nice day.